Étienne, and I'll talk to you today about SnoBIRD, a tool I developed to predict CD-bug snoRNA genes across any eukaryote genome. So snoRNA stands for small nuclear RNAs. These are a type of non-coding RNA present in all eukaryotes. Their main function is to guide modifications on a target RNA to which they bind, and they are also involved in the regulation of a lot of different processes such as splicing and even pre-mRNA stability. So what we found in a previous study is that when looking at different RNA-seq datasets in human, when looking at the expression of these snoRNAs in these datasets, only 1/3 of all annotated snoRNA genes were expressed, and 2/3 were never expressed across all of these healthy human tissues. Again, when looking at RNA-seq datasets in mouse, we saw a similar trend. 1/3 that were expressed, and 2/3 that were never expressed. Now, we wanted to better understand why some snoRNA genes were expressed and others were not. So we trained different machine learning models on different features of these snoRNAs and interpreted their decisions to find on which features did they rely the most to make their predictions, basically telling us which feature were important to regulate the expression of snoRNAs. What we found was that the main feature regulating whether snoRNA was expressed or not was the box conservation. So whether the boxes or motifs here in red were conserved or close to their consensus sequence, which was really the case for the expressed snoRNAs here on top, and was really less the case for the non-expressed snoRNA genes, which accumulated a lot of mutations within their boxes. So we decided to call these non-expressed snoRNAs snoRNA pseudogene because of these difference between these two types of snoRNAs. Second big feature was that these snoRNAs were a lot more stable in terms of structure compared to these ones. And the third one was that there was this structure here called the terminal strand that was way longer and also way more stable in expressed snoRNAs compared to snoRNA pseudogenes. So why should we care about distinguishing these two types of snoRNAs? So there's this little analogy here that I present. So on top you have this highly skilled feline, which is the lion, which is really successful at hunting in the wild, and it would compare to this functional snoRNA that's really helpful or functional in the cell. And there's also this other type of feline, which is the cat here, but that is way less dangerous and mostly harmless, that could compare to this other type of snoRNA that is most likely not functional. So basically the reason why we wanted to distinguish these two types of snoRNA is because they have completely different functionalities, and right now there is no method that can actually differentiate between these two types of snoRNAs. So I built SnoBIRD, which is a machine learning model based on BERT models. And to train that model, I had to collect a lot of different examples. So examples of expressed cdsNRNAs, snoRNA pseudogenes, and also different negative examples, which were picked by randomly picking actually regions in the genome of different species, and also by taking other types of negative control non-coding RNAs such as tRNAs and snRNAs. So I collected these data from RNA-seq datasets across all eukaryote kingdoms, so animals, protists, fungi, and plants. And all of these examples were selected based on the sequence only of the snoRNA. So this sequence was fed as input to SnoBIRD, which was then chimerized into little chimers of 6 nucleotides. This was fed to an embedding layer, which was then fed to our pre-trained BERT model. And then this pre-trained BERT model was fine-tuned on our dataset to predict our first classification step. So whether within that sequence it contains a cdsNRNA gene in the general sense or something else. If it contained a cdsNRNA gene, we then trained a second model, a second BERT model, as well as apply different features based on the snoRNA feature, filters based on the snoRNA features, which So the second classification step told us whether that CD-snoRNA gene is expressed or CD pseudogene. So I'm presenting here different results. I won't go into big details of all, just here on top you have the performance of SnoBIRD compared to its competitors in predicting CD-snoRNA genes in a general sense on the test set. So you can see that it's the best or even second best on all of the metrics that are represented here on the x-axis compared to the other tools. Interestingly, when we applied SnoBIRD to a genome that was never seen during the test or even the training, which is the fission yeast S-POMB, we could predict with SnoBIRD a lot of different candidate snoRNAs that were actually expressed and that were never annotated before and not even predicted by the other tools. For instance, this snoRNA here that is highly expressed, even more expressed than actually annotated snoRNAs, and we can clearly see its distinct motifs, so the C and D boxes, which are actually really important in the prediction. So that's what's presented here. You have the SHAP values on the y-axis and the x-axis, the sequence, and you can see that there's two peaks of importance that are, so these parts, the C and D boxes, are used by the model. So it really relies on biological signal to make its predictions. So with that, I'd like to remind you that I created SnoBIRD, a CD-Box snoRNA predictor tool that can be applied to any eukaryote and that can differentiate between expressed and snoRNA pseudogene. And that's it. Thank you for listening.